We're very pleased to honor you with as a recipient of the 6th Annual Maria Luisa Ferrari Lectureship for Life Award. And just wanted to ask you a few questions about your view of cancer research and how that may go forward. You have directed your research now in the carbon chemistry in the direction toward cancer research, and I just wanted to know how you feel that will go forward and your vision for the future of cancer research in general.
Well, my research has for years been focused on things we call the fullerenes, of which the prototypical example is a ball. Let's see, it is in fact exactly the same pattern of seams that you have in a soccer ball, except this is made out of carbon and it's only a billionth of a meter in diameter, called a buckyball. It turns out to be the first member of an infinite series of new materials we call the fullerenes, of which by far the most interesting are elongated versions of the ball with two end caps on either side and connected by a tube of pure carbon, which we lovingly call Bucky tubes. And we've been heavily involved in this research for over 15 years now. And one of the areas of applications of these tubes, and for that matter the balls, is in the diagnosis and treatment of diseases in general and cancer in particular. I've been alerted to this issue by the fact that about 7 years ago I myself was diagnosed with leukemia, lymphoma, and so my way of coping with this has been to learn as much as I possibly could about these diseases and the molecular biology of what's really going on. And so that's happened sort of simultaneously with the development of in my research group of these Bucky balls and Bucky tubes and what is really molecular and materials physics and nanotechnology. And as the years have gone by, we begin to realize that there are really stunning possibilities for the use of these tubes and the balls in the diagnosis and treatment of diseases such as cancer, both inside the body and outside. So it's really been quite a fascinating journey from my viewpoint. You know, you often say that life is something that happens while you're busy doing something else. And that's certainly been the case for me. While we're busy working with Bucky tubes, this cancer thing has alerted me to the entire world of applications and the subtleties involved in multicellular organisms when they get diseased in cases like cancer.
That is fascinating. Where do you see the cancer research going from here?
Well, as most of you know, cancer is an incredibly complex disease. Probably there are hundreds, if not thousands, of different types of cancers, and the mechanisms of this disease reach to the very essence of what it is that makes cells function and interact with one another, and not only side by side a few cells, but whole organisms, pretty much the whole body. So it would be hard to find a more complex disease. I live about a mile and a half away from the MD Anderson Cancer Center, which is the largest cancer center in the world. Their motto is making cancer history, and they fully intend to accomplish that for most cancers within the next 10 to 20 years, and I rather suspect that's going to happen, not just with people at MD Anderson, but with worldwide efforts of scientists worldwide as we not only begin to understand this disease, these diseases in fascinating details, but with the incredible new technology that's coming along, both with traditional biomolecules like DNA and RNA and so forth, but also with new molecules, new things from what I call the dry side of nanotechnology, things like Bucky tubes, that are themselves molecules. They're like biomolecules, nanometer in scale. But they bring things new to the game, like in the case of Bucky tubes electrical conductivity, the ability to interact with electromagnetic radiation, all the way from radio waves up to gamma rays, for that matter. So we're trying to bring some new things, some new gadgets to the table as we look for ways of controlling this disease and ultimately eliminating it. I think most of us believe the best way to cure cancer is not to get it in the first place. And if you do get it, which almost certainly you will, some cells will start to go through the sort of transitions that are the precursors of a malignant cancer, to detect that very early on and to treat it very early on with great knowledge of just how many such cells you have in your body and whether or not that treatment is effective in eliminating them. Cancer is very much of a numbers game. At one time, I had on the order of 10 to the 13th cancer cells in my body just circulating around, and probably a little bit more of that in the lymph nodes and the bone marrow. And we knocked it back by many orders of magnitude, so we couldn't detect it anymore. We're really quite proud of that. We call it molecular remission. And yet, when you really look at how well we could detect these things, there could very well have been 100,000 cancer cells still in my body. And in order to cure cancer, you got to get down to that last cell. So it's a numbers game. And we're still at a pretty crude level at being able to detect these things at the single cell level. And it is in that detection that we have the greatest potential for the new nanotechnologies to have an impact on this disease in the next 10 years or so. For example, imagine that it were possible to go to a clinic and for $50 get a blood test taken. But instead of, with the current blood test, you have the concentrations of something like 20 things in your blood that is being measured. 10 years from now, it's 20,000 things that are being measured, including trace proteins and snippets of RNA and DNA that are circulating in your blood system, which goes everywhere through your body. And by looking at the concentrations and identities of this 20,000 somehow species, you have a snapshot of what's going on in that body. And you can compare it with what that was 6 months before. And most cells, when they go into a disease state, either by rational mechanisms or just by accident, release to the peripheral blood proteins in various concentrations, little snippets of other molecules, which, if you could detect them in trace amounts, will give you a diagnostic that, in this organism there are some cells that are either cancerous now or precancerous, and you get some sort of quantitative indication. Just think how powerful that would be, because very early on, in many cases, we believe just a change in your lifestyle would have a dramatic effect. Eat broccoli. Well, how do you know you're having an effect? This blood test would be great. Well, how are you going to detect 20,000 things in